Good afternoon, everyone. My name is Andrea Cavalli. I'm director of CECAM. CECAM is one of the partners of the MDDB project. The MDDB project is a project funded by the European Union dedicated to the creation of the first unified database for molecular dynamics in biological system. Proteins, RNA, DNA, membrane, with the objective to create something similar to the protein data bank for dynamical simulation, conserving and collecting all the trajectories produced worldwide in the field of molecular dynamics applied to biological system. The final objective is to create a global repository of these trajectories, which then can be utilized for many different applications. Just to give you an example, we increasingly know that drug discovery targeting proteins and targeting nucleic acids may benefit from the knowledge that we can gain from molecular dynamics simulations. And in particular, the possibility to identify novel pockets where innovative drugs can bind is one of the big challenges in computational drug discovery and computational medicine. Without any further delay, I would like to present today's speaker, Dr. Andrew Emerson, application specialist in Italy, working at CINECA in Bologna. CINECA is one of the high-performance computing centers contributing to the MDDB effort. Indeed, MDDB will need collaboration with supercomputing center for building the network of databases that then will be available for the entire scientific community. Today, Andrew will talk about high throughput molecular dynamics simulation in LIGATE and how these molecular dynamics may impact the future of computational biology, computational drug discovery, and more in general biophysics. Please, Andrew, thank you very much for accepting our invitation to deliver this webinar, and the stage is yours.
Thank you very much, Andrea. Thank you very much for the introduction, and thank you very much for giving me this opportunity to demonstrate some of the work we did in the LIGATE project. Just a little bit of information on where I work. So I'm at CINECA, which is Italy's largest supercomputer center. It's based just outside Bologna in Casalecchio di Reno, but we also have offices in other parts of Italy, including Milan, Rome, Naples, Chieti, and also a new one in Sicily, in Palermo. So this is sort of a photo of the outside of the head office. The CINECA's main facility, though, called Leonardo, is based in Bologna, in actual fact, at the so-called Bologna Technopole, together with so the ECMWF Weather Center and various other facilities here. So this is a major facility which is based in Bologna. Leonardo itself is, I said, our major supercomputer. In the top 500 list of supercomputers in the world, it is currently number 9, but it did enter in number 4. So it's one of the most powerful computers in the world and of course in Europe. What I want to talk about, so to lead up to how we our collaboration with MDDB, it starts actually from the topic of drug design. I think you know all this, that new drugs are very expensive, there are long lead times, 10 years, and so on. And it's a very difficult, expensive thing to do. And what we want to do is to use computer-aided drug design to make it to try and push this curve up here, to make more drugs available more quickly. The project I'm going to talk about started during COVID era, and well, as I think you're well aware, we didn't have any good therapeutic drugs for the virus, or for any viruses in actual fact. So this is the LIGATE project. I don't want to go into detail about this because for some parts are probably not going to be interesting to this audience. And in any case, it finished at the end, well, towards the middle of last year. But this was a very interesting project from many points of view because it wasn't a scientific-based initiative, but it was more based on practical, actually trying to get software and programs and workflows and so on, working on the architecture, the various architectures that are in the world right now. So I mean, there's lots of interesting things in here. So apart from GROMACS and so on, there's also something like the Lexis workflow engine, which is going to become part of the UHPC Federation drive. We looked at management of large data sets and so on. The main focal point of this project, though, was here, so centering on virtual screening to find new drug compounds. So again, you know all this stuff, that it's not rocket science, but the idea of high throughput virtual screening is to able to somehow filter a very large space of molecules or molecule fragments in such a way that we can find those molecules most likely to be of interest. So, for example, we may be looking at a target, a viral enzyme, and we want to filter all the possible chemical entities and find only those most likely to be effective, to bind to the enzyme. So this is the idea of this virtual screening here. And you think of a very simple model here, the lock and key, that you try and find something that fits into the cavities on the surface of the protein. The ligand software, the virtual screening software we were using is something called LIGATE. This is a software developed in collaboration with Dompé Pharmaceuticals, a pharmaceutical company in Italy, together with CINECA and other partners. The point about LIGATE is it's very computationally efficient. So, I mean, my colleagues who are expert parallel programmers have made a very efficient, helped to develop a very efficient system which can run on today's and hopefully the future's supercomputers. So a bit of background here. So in this top here, this is a diagram taken from a sister project of LIGATE called Escalate for COV. This started right at the beginning of the COVID period. And using a virtual screening experiment with LIGATE, we actually managed to identify a drug, a generic drug used for osteoporosis. And actually, this passed phase 2 clinical trials for COVID. In a more sort of, I don't know, brute force demonstration, we managed to run a very large virtual screening experiment, which we call the Big Run, on 2 computers combined together. So in this case, it was the M100, the predecessor to Leonardo at CINECA, and the NE HPC5. So this was another large computer. And well, I put the numbers there. I don't know if this is the largest run ever performed, but it must rank up there. So this was a very massive effort. Okay, but there's a problem in all of this, isn't there? So, I mean, there's a fly in the ointment, so to speak, because, there are various ways we can look for for where a ligand binds on the target protein and so on. But what we really don't know what to do is is how to score, how to rank the ligands. I mean, particularly in high throughput mode, where we can't spend more than, let's say, a few milliseconds on each docking event. So, I mean, how do we rank the ligands? What's the best way of doing this? And this has been a subject of discussion for a long time. So I think at the end of my PhD, quite a long time ago, 30 years ago, my colleagues involved in biomolecular simulations were asking about this then, and it's something that we haven't solved. So this way of ranking the ligands is called the scoring function, and this is where this is sort of one of the main focal points of the LIGATE project. So the list of scoring functions, there are many. You can sort of divide them in sort of two, if you like, two groups, the classical scoring functions. So these can be sort of energy physics based, you calculate interactions. You can have some sort of empirical method, other based on knowledge of the ligand and protein and so on. And the various docking programs have their own mixtures of these types of scoring function. Then there's the machine learning based. I'm not a really big expert on this from the papers I read. Until recently, let's say, there was not so, let's say, successful. So the problem you get with sort of AI sort of things that you sometimes end up with non-physical sort of interactions. In LIGATE, we tested AlphaFold to see if we could use it somehow in virtual screening, but it didn't work very well. We found that it didn't give accurate sidechain modeling, or accurate enough for our purposes. So one of the big aims of LIGATE was to find an improved machine learning model. Now, okay, there's a space in this slide here, so I'll explain why. So I think, as you probably all know, around May last year, AlphaFold 3 came along, and the papers will say the same thing, as there's a massive increase in accuracy. This is an improved machine learning model based on the diffusive type model, also used in generative AI. The problem, though, of course, is that unlike AlphaFold 2, which you can download and install, AlphaFold 3 you can't, and the license explicitly prohibits the use of HTBS, so high throughput screen. Anyway, let's go back to our problem. So how are we going to create a machine learning model for high screening experiments? So I'm going to make a couple of assumptions here. The first one is, this is what the assumptions were made in the project, is that ligands can be ranked by the absolute binding free energy of the protein and the ligand. So if we had a very fast way of calculating the free energy of the protein and the ligand, we're assuming this could be a good way of ranking of the ligands. I think this is reasonable, so I don't think anybody would disagree this is not a bad idea. The second assumption was, okay, we shall see whether it works or not, but the idea was that instead of taking experimental data directly from some database, we would use molecular dynamics instead and get the data from the molecular dynamics. The rationale behind this was that we wanted consistent data. So if you look at the literature for how free energies are calculated from equilibrium constants and so on, various methods are done, and with various protocols. And if you're doing a machine learning model, this is not ideal, because you want a consistency in your training data. So we decided to use molecular dynamics to do this. In theory, you can run a molecular dynamic simulation, and it should be possible to calculate the energy, this absolute binding free energy. But in practice, it isn't. It doesn't work. And And well, the AMBER website has a good explanation of all this, but basically the fluctuations in the solvent free energy just swamp your signal. It doesn't work. So what you have to do instead is to construct a free energy cycle, and then you can get the term you want by calculating the other terms. Now, this is quite a big subject here, but you have to make quite a few assumptions. So the assumptions are so-called single trajectory protocol, where we use a technique called MMPBSA. Yeah, you assume a lot of things. You assume rigid docking, no changes of structure between ligand and protein. You also have to assume that the entropy terms cancel out and so on. There's quite a lot of assumptions. But the basic idea is that you can run a normal MD simulation and then pre-process it to get the free energies. So let's look how we do this. So you do an explicit solvent MD on which you get the trajectory. You remove the water molecules, and then using snapshots from the trajectory and using an implicit solvent model, you can calculate the free energy. The question here arises in, well, how do we model the implicit solvent? I'm a bit out of my comfort zone here because this was done by other colleagues in the project. But in any case, we looked at 3 different methods, the so-called MMPBSA based on the Poisson-Boltzmann calculation, a simpler version called the generalized Born model, and another one called 3DSIM. Yeah, I can't discuss this anymore because I don't know the details. But anyway, we tried these 3 different techniques. And we tried it on a validation set. So I should have pointed out earlier, we were using a set of protein ligand pairs from a database called PDB-Bind, because this also contains experimental data. And in fact, they have a refined set of about 5,000 complexes, since they have been trained fairly successfully with machine learning models. So in our validation set of about 60, 70 complexes, we calculated the 3 energies based on these 3 methods. And thankfully, well, thankfully, conveniently, the simpler GBSA method gives the best correlation. To do this, you have to make one minor alteration to the input parameters of the MMPBSA program. So we changed the dielectric permittivity, but otherwise, we get best results from GBSA. What was also quite, I gave a big sigh of relief when my colleagues came up with this. They decided that 250 nanosecond simulation would be sufficient. Now, I gave a sigh of relief because the idea of doing 5,000 simulations, well, 5,000 4 replicas, each for 1 microsecond, for example, it's sort of, made me a bit nervous. So, I mean, it's one thing doing 1 microsecond simulation, which we have to do 16,000, 20,000 of them or something. It starts to become, rather complex, also because you need a lot of GPU time. But thankfully, we set it on 250 nanoseconds. So that was good news from the point of view of the MD team. So what is the workflow like? So we start off with this refined set of just over 5,000 complexes. The team from KTH then ran their workflow to generate the GROMACS topologies. And unfortunately, yeah, some complexes were lost on the way. So the thing is, when you start doing things in high throughput, you can't check every time something goes wrong. So now you can modify your workflow a bit, but in the end you have to, assuming, you've got, in this case, we've got about 80% to 90% successful topologies, then we leave it at that. So we can't check for each complex of which can give a topology. Then the usual MD sort of preparation, minimization, equilibration, production. So we, as I will show later, we did around 1 complex per GPU and 4 replicas per complex on Leonardo. Then there was a period of trajectory post-processing, archiving on a different computer called Galileo 100. So this is because the archive that we have at CINECA is on this machine. And then we have this part here where we run the MMPBSA program to calculate the free energies, and the idea this goes on to the machine learning model. Give a few more details here. So this is the workflow here. So yeah, as you can see, as we go along the chain here, we sort of lose simulations as we go around. So not all the topologies can be made, as I said earlier. And then we weren't able to simulate all the topologies either. I go into this a bit, but yeah, we had problems with equilibration and so on. And obviously some complexes also failed during production. Again, if there had been large numbers of failures, we would have to look very carefully, but given we had to do this in about 4 months, we couldn't check every time a complex failed, where we sort of sampled the failed simulations and went ahead. So where did we run the systems for the simulations? We ran this on Leonardo. This has been jointly funded by UHPC and CINECA. The important part from the point of view of the dynamics is the booster module here, which is 3,500 nodes each with 4 A100 GPUs. This is the sort of node diagram here, and this is the node, the physical picture of the node. I didn't go into this, but to get the computer with so many GPUs to work for a reasonable amount of energy, you have to liquid cool directly onto the chips. So these copper tubes here actually contain water, in fact, warm water, in order to cool the chips down. So if you try to air condition the booster of Leonardo, I mean, the energy bill just would be unsustainable. How do we run GROMACS? So this is something I would like to emphasize a bit, that except in some rare occasions, you don't need to use multiple GPUs or MPI to run a molecular dynamics simulation. This follows for both GROMACS and AMBER. This is because now these programs can stay almost entirely on the GPU. So we don't have to even worry about CPU, GPU connections anymore, really. These simulations can stay entirely on the GPU. You have to be a bit careful that you decide what's going on the GPU, but this is just one simulation, one GPU. So that there is no need to worry about scaling curves or anything. In actual fact, what we could have tried, but didn't, we could have even tried running multiple simulations on the same GPU. So that would have increased throughput further. But anyway, so in this workflow, one simulation, one GPU, and that's it. You've got to avoid printing out too often, because this causes a transfer to the CPU. But this gives very good efficiency. Something to take account of when you do simulations, at least with GROMACS, I haven't checked with other packages, you have to check something called process pinning. So in process pinning, even though the simulations run on the GPU, they are still associated with sets of cores on the CPU. And what you don't want is processes migrating over the CPU You want this GPU always associated with these 4 cores here, these 8 cores here. And this in GROMACS you do with this this pin on option. And by carefully selecting the pin offset, pin stride, and so on, you can make sure that this GPU only contacts these cores, this one and these ones, and these as well. If you get this wrong, you can get very bad performance, as we found out in the previous Marconi 100 computer, where the numbers here were different. So we got very wide varying performances. So, you know, this is something you should take care of. What about the workflow? How did we manage that? So in retrospect, if I had more time, I think I would have selected a proper workflow manager, because, you know, with one simulation, it's fine. You just sort of run it and analyze it. But when you're running 16,000, and also with team members as well, you got to make sure that, you know, the simulations are managed properly. I had a look a bit at, you know, some of the bioinformatics workflows or things like SNAKEMake. But given that some of these simulations can take up to 2 weeks, it's not quite straightforward how to do that. In the end, we developed a sort of Python-based workflow, which seems to work. But I think if I had this in future, I would look into this more deeply. What's the results we get? Well, okay, so we didn't really analyze the molecular dynamics simulations themselves, because our idea was to put them into a machine learning model. But just a couple of slides of results. So performance this axis, number of atoms this axis. And as you can see, I mean, even just using 1 GPU per run here, you can end up with, you know, 1 microsecond a day, basically, for the smaller complexes, which I still think is quite remarkable, considering, you know, 10 years ago, we used to, you know, talk about simulations of a couple of 100 nanoseconds. So the fact that we can do this in 1 day now is amazing. Just out of curiosity, I also did this sort of fitting here, this 1/x fits very quickly. And as you can see, it works fairly well up to about 200,000 atoms. So in other words, you know, we really do get this sort of 1/n scaling law. It falls off a bit here, but on the other hand, we sort of run out of points here as well. But I actually quite like this result, because, you know, you see in the textbooks that you should get this roughly sort of scaling law. And, you know, with 4,000 points here, we've sort of demonstrated it. So it means at least that, you know, for this sort of these complexes, if you know roughly the number of atoms, you can guess roughly what the performance is going to be, regardless of the nature of the protein or the ligand. Trajectory analysis, as I said, we didn't do this really in the project. In fact, I did this last Friday. Yeah, some of the simulations, they do show this association during the trajectory. So in actual fact, in the post-processing stage, we looked for 3 checks. We call this protein stability. Probably flexibility is a better word. I don't know, protein stability, ligand dissociation, and ion dissociation. Ions probably in the binding side. and yes, probably about 1/3 of the simulations dissociate in at least 1 replica over the 250 nanoseconds time scale. And, well, many of these in a very short period of time. I was a bit worried about this during the project. And as in fact, to get decent free energies, you only need a few nanoseconds. And in any case, this is, you know, this doesn't happen very often in all 4 replicas, but still, this influences should influence the results you get in the machine learning model. Obviously, you have to leave them in there, because this is, you know, otherwise you would bias your results. A few pictures here of other things we saw. It's useful having, you know, this sort of data in a database, because you can do a lookup and say, I don't know, find me a complex with which is highly flexible. Find me a ligand which leaves after 100 nanoseconds and so on. So it's a very convenient thing you can do. Now, a few issues. So there are a few issues, of course. Of course, the point is when you do things in high throughput, you can't check each simulation which fails. You can only sort of sample perhaps or investigate major faults. So we had for some larger complexes over about 200,000 atoms, I suppose, we were getting 100% failure rates, which was a bit disturbing. But after performing various, different sorts of equilibrations, particularly pressure coupling, we managed to reduce this. Some things you just can't reduce entirely, also because they may be due to hardware. I mean, once you start having very large computer systems, some nodes are going to be down or faulty in some ways. Some analysis routines failed as well. I don't know why, but it wasn't serious. So what about machine learning? How did that go? How did the results go? So this is the part of the presentation I know least because it was done by colleagues. We use this sort of machine learning model here, based on this article here. Yeah, I can, if anybody asks any questions about it, I can forward to the correct people. Disappointingly, it didn't work very well, unfortunately. So when you're analyzing the results of the virtual screenings, you have a number of these metrics described in this table here. And so these are results here, this first line here. And this second line here, we used the same complexes, but we took the energies, the experimental energies, and here we ran the whole PDBind 2020 set, nearly 20,000 complexes here. And actually this gave the best result. So after all this effort, in actual fact, the MD approach did not give the best results. Why? Well, the obvious one is a small data set. Machine learning, the size of data is really important. But there's lots of other possibilities as well. I mean, in our approach, we ignored conformational entropy. We use a single permittivity value and all sorts of things like that. But anyway, this is, in principle, you could try and do better than this, collect more complexes and so on. But the number of complexes is clearly an issue. Here's some of the resources we used. It was about 800,000 GPU hours donated by CINECA. We have about 40 terabytes of archive space for the trajectories. And if anybody's interested in learning more, I have put the resources there. So a few lessons learned. I'm not saying you get maximum performance with one GPU, but one GPU really is enough in most cases. And although I was, not sleeping for a little while, wondering if I'd ever get these thousands of simulations done, in practice you do. Now, this is the bit I want to concentrate now, and the reason of this seminar is that the project finished, but we still want to keep the data. We still want to share the data. The machine learning model didn't work very well, but we still have over 16,000 ligand protein complexes, which we would like to keep. Currently, it's sitting on some disks in CINECA, but apart from the fact it's difficult to share, this is not a very good way of keeping it. And as I think as many of you know, in Italy and Spain last year, there was severe flooding, which affected, which was very serious. So I mean, in Valencia, many people died. But even in Emilia-Romagna, for the second time in 2 years, the large areas in the Emilia-Romagna region, including Bologna, were flooded. And this included also the machine room of CINECA. So when this happened, I was crossing my fingers, to see if the data would still be there when the flood waters, subsided. Apparently the data was, it's only really become stable now, the disk system. I'm extremely happy then that Nani Beltran and Adam Hospital came to CINECA to give us this MD seminar, and they set up the server that we have at CINECA here at the moment. I must say it's very impressive in many ways, also because we didn't have time to look in onto these all these things here during the project. We didn't have time to calculate all the RS and RMSDs, hydrogen bonds, and so on. What we need to do is to upload all the complexes into the CINECA MDDB. At the moment, there's only 33, unfortunately, but hopefully within the next few months, my colleagues and I will be able to upload this data and, finally keep it safe, so because at the moment, sitting on a disk is not the ideal thing. So not only safe, but also be able to share it with anybody who can find it useful. So that's the end of my talk. Acknowledgements to a lot of people, particularly the CINECA MD team, Georgia, Andrea, and Lara, the KTH people, Akash, Domenico, who did a lot of work in this workflow, LIGATE Consortium, and big thanks to MDDB for giving us a chance to keep this data alive. And thanks again to the UHPC for funding this work. And that is the end of my talk. Thank you very much for your attention.
Thank you very much, Andrew, for the very insightful presentation concerning the use of molecular dynamics, particularly in the field of ligand identifications for pretty dangerous pathological conditions that we experienced over the last few years. There are some questions for you in the Q&A, so please feel free to add more questions if you have any comments or clarifications you may need. The first one is from Matthias. Which program did you use for the generation of topology file? GMX to top or GGenf?
That's what, yeah, we used, okay, so this was done by KTH. So we used, so the topologies were GROMACS topologies, but there's quite a long workflow here of, checking if the protein needs repairing, if it can be patched, and then have the force field and so on. So there's a long chain of other programs involved here. I don't know which ones in particular we used. I can check if you like, but there's a sort of mini workflow of various programs to, in an automatic way, convert the proteins to the topologies. We can't manage each one by one, so they all pass through this sort of workflow.
Thank you very much, Valentin. Technical, and which is one of the most interesting questions for the time being. When running MD, particularly on GPU, and we know that there is the CPU bottleneck, how much this impact on your simulation, which is the load balance between CPU and GPU?
It's now, if you run GROMACS or even AMBER in GPU resident mode, it stays almost entirely on the GPU. So the connection CPU and GPU is not a bottleneck because it's entirely there. So this is the amazing thing about it. You can make almost every single calculation that GROMACS does on the GPU. I said the only thing, I think in my slides, the only thing you have to worry about is if you print out energies very frequently, because then has to come back to the CPU. So, I mean, if you start calculating or start printing out energies every time step or something, that's going to degrade performance a lot. But if you do it every couple of hundred of steps, you know, that's fine. If you do it in this way, it all stays on the GPU. It's really very impressive. So it's a lot of work has been done on it.
Again, Valentin, do you have any thoughts on the dataset enrichment through generative AI methods? Probably at that time, it was not something very, very well established as a field. And more in general, will you still recommend to use physics-based methods, or
so again, this is a bit out of my comfort zone. Certainly a problem with the approach was, well, there were two. The big one was the lack of complexes, the lack of the data set. So if there was another way of enriching the data set by whatever method, yes, it could be interesting. There are also obviously simplifications in the model. So I mean, we didn't take account of configurational entropy. We've seen that in some other programs, you know, they use a different, you know, variable dielectric constant and all this sort of thing. From a machine learning point of view, I don't know what the best way to proceed with this is. I really don't know. But yeah, clearly the data set was too small though.
Yeah, I think that there is another question here, Georges. Thank you for the nice presentation. If we use a rigorous thermodynamic approach for the free energy extraction, for example, free energy perturbation or alchemical transformation, will we obtain better results in screening, taking into consideration both the accuracy and computational cost? Are there efforts in this direction? So if are you planning to refine your outcomes with free energy calculations, Andrew? Or
so the project is finished and I'm no longer working on this. But the thing is, though, I mean, if once you start doing more accurate methods, then of course the time starts to become important. So also in virtual screening, you want something which is done extremely quickly. This is why we wanted to use the machine learning model. I don't know if, I mean, surely you get more accurate free energy with more, you know, accurate methods like free energy perturbation. But I mean, can you do this on thousands of molecules? I don't know. So I mean, you know, it's and with high, I mean, even the the virtual screening. There's I mean, even the rigid protein, the rigid ligand. There's a large number of approximations there. It may well be that it's a bit surprising that the GBSA method, for example, gave us better results than the more sophisticated PBSA method. But that may be because we didn't have time to adjust all the parameters in PBSA, for example. So I don't know if anybody in the consortium is continuing with this line of work actually. So I don't know.
Well, we know that free energy calculations can be very, very demanding from the computational standpoint. Probably in the lead optimization or in the heat-to-lead running FEP can be still doable, particularly now that FEP has been optimized for GPUs. Still, I think not in the order of thousands of compounds. I think in the order of few hundreds of compounds. And in these conditions, probably FEP or other methods based on thermodynamic integration or more in general chemical transformation can provide much more reliable outcomes, even though the number of molecules that you can process is certainly limited. And
yes, I mean, you get better free energies, but yeah, I mean, it's certainly the number of complexes affected the precision of the machine learning model. Machine learning model, of course, you need to have chemically diverse structures so as not to bias the data set. So yeah, I don't know. I think unless you have an accurate method which is also can process thousands and thousands of molecules, yeah, it's going to be difficult.
Yeah. Another question is from Tanya, exactly in the spirit of what MDDB is planning to do. Trajectories are available on the node repository of LIGATE of the LIGATE.
Yeah, we can't put 40 terabytes on Zenodo.
Okay.
So they are available at CINECA, but we said we are planning to upload them to MDDB as soon as we can.
Okay. Okay.
We sort of got delayed also because of the the events in Emilia-Romagna last year. So the data is there, but the sort of best disk systems sort of became unstable. The idea is to upload as much as we can to MDDB. So there's 16,000 of them, so it will take a while, I suppose. But the idea is to put everything there. If you have access to CINECA somehow, then you can access them directly from there as well.
So for the time being, there are two possibilities. Either ask you the trajectories, and then you can try to give them to Tanya and anyone else, or wait when they will be available on the MDDB project and database, which will happen in the next few months.
The next few months, yeah.
Few months.
I'm hoping to, I've got a final deadline for keeping the data there because we don't have long-term data storage for years, so my plan is to get it there by July. If somebody's interested in a subset of the data, I can transfer to Leonardo, for example, and they can look at it there. It's possible to get access to Leonardo, even if you're not Italian, by UHPC. So in principle, you could ask for a UHPC project, and I can make a subset of the data available there. If you're interested, just let me know. But the idea is to have it in MDDB, by the middle of this year.
Did you have a general curiosity also in the spirit again of MDDB? Did you analyze this amount of data, large amount of trajectories that you generated from the critical transient pockets standpoint, trying to identify a number of pockets for each single protein?
No, we didn't do that. No, because we took so this was another part of the LIGATE project, but it wasn't part of this workflow. So I mean, we took protein ligand complexes already in their complex state, shall we say, from the PDB-bind data set. So we didn't do further analysis of this. This could be interesting, actually. This could be a very interesting thing to do.
And again, this is in the spirit of MDDB. Once the trajectories will be available, anyone can apply, for example,
absolutely,
pocket detection tools, which is a not topic in drug discovery, because to identify allosteric pockets or more in general cryptic pockets in proteins is one of the major efforts to identify very selective drugs. And
so we sort of did this in other parts of the project, but in this particular workflow, no, we didn't. But I think this could be very interesting use of the data.
Okay. Apparently, there are no other questions. There were more than 50 participants, so I'm very satisfied about your the number of people coming to listen your presentation and your lecture. I thank you again, Andrew, very much for delivering this seminar, this webinar, and I leave the stage to our admin, to Aude, for some information about the future webinars in the context of the MDDB events. Please, Aude.
Hi, so you will see on the screen now our upcoming MDDB events. We have another webinars session on February 21st with Yasaman Karami. All the information will be published on the MDDB and CECAM website, so please keep an eye out for the Zoom link for this webinar as well. A fourth session will take place in March. The date is still to be confirmed. Again, you will find all the necessary information on our social media and websites. And in October, there is a workshop open to the community titled When Data Science Meets Molecular Dynamics, and registrations are open. You can find all necessary information on the CICAM website in our program in upcoming events. You can submit your application and attend the event here in person in Lausanne. Thank you.
Thank you very much, Aude. Thank you again, Andrew, for the webinar, and thank you to the participants to this session, and see you on February 21st. Thank you again. Bye-bye, and a good afternoon.
Bye. Thank you.
Thank you very much. Bye-bye.